we're very excited here at Iowa State to be able to describe and announce the sequencing of the corn genome. This is a historic achievement. It's something that scientists have been working for the last several decades to achieve. They've been laying the groundwork, developing the technologies, and starting 4 years ago, the National Science Foundation invested $30 million in a group of universities, including Iowa State, to sequence the corn genome. It is, though, the most complex genome that has been sequenced to date. Plants, like animals and bacteria, contain DNA. DNA is the basic genetic material that tells the organism how to grow and develop. All of the DNA within an organism is called the genome. Mapping a genome is trying to identify, to determine where the genes are located on the chromosomes. Sequencing the genome is going beyond just finding out where the genes are, but making sure we know what all the genes are and then characterizing them with enough information so we can make educated guesses in terms of their function. Genomes are sequenced traditionally, that means 4 or 5 years ago. Genomes were sequenced by first taking the DNA and chopping it into medium-sized bytes. And then those medium-sized bytes of genome were chopped up into little tiny nibbles, and we could determine the sequence of these little tiny niblets of the genome. And then using computers, all of that is put together again. It's like putting together a giant jigsaw puzzle where a lot of the pieces look very similar and some of them have no pattern on them, so it's very hard to figure out which pieces go together. Mapping the genome, sequencing the genome, gives us very important clues as to how the corn plant grows and develops. With that information, we can improve traditional plant breeding practices as well as develop and modify the corn plant to meet new human needs. It's also it's defining the various biochemical pathways in the corn plant, which will suggest new ways to adapt corn for new uses. So right now we use a lot of corn for food and feed and fiber, and more recently for fuel. But we can also use corn as a raw material for industrial products. We're entering an era where we need to have renewable products that go into our industrial programs. And so we can re-engineer the corn plant to produce raw materials for our industries. And the corn genome sequence helps us do that. Another aspect of this project, I've mentioned that putting the genome together is like solving a giant jigsaw puzzle. And we've been very fortunate to have Schnable as a collaborator on this project. His expertise is high-performance parallel computing. And through investments of both the state of Iowa and the National Science Foundation, we have some very large-scale computers here at Iowa State that have helped us understand the structure of the corn genome. Schnable and I are both members of Iowa State's Plant Sciences Institute. So the Plant Sciences Institute is an umbrella organization that brings together faculty from different departments, different disciplines, and helps enable them to work together in very productive ways that reach across the traditional boundaries of scientific disciplines. And so this project is actually a prime example of what PSI is quite good at, which is bringing people together who might not work together normally to do something bigger and better than they might normally do.